human biology students, we're in lab 8, which is blood. And for this week, you have a worksheet to do again by Sunday, as well as last week's quiz. Here are some learning outcomes identifying. We'll look at the components of blood, their functions. We'll look at some common blood tests. We'll know what determines blood type and the importance of understanding your blood type. We'll apply the knowledge of basic anatomy and physiology through laboratory investigation that we have kind of through these lab guides. So again, use the lab guide as you're working through the worksheet. Blood itself is a specialized type of connective tissue. It includes formed elements, and the formed elements are erythrocytes, which are red blood cells, leukocytes, white blood cells, and thrombocytes, which form platelets to help blood clotting. The red blood cells, white blood cells, and platelets, again, which are the formed elements, are all carried what we call plasma, which is the yellowish fluid portion of blood. Plasma can also contain some nutrients, proteins, hormones, and antibodies produced by a type of white blood cell to be discussed later. So if we look at kind of a picture of your blood, the majority of a blood sample in a microscope slide would show the erythrocytes. You have 5 different types of leukocytes, which will look a little different. And then the thrombocytes are little tiny dots that will go on to form the platelets, which help to clot your blood. In the first exercise, we'll use a microscope to view a prepared slide in order to illustrate a blood sample, complete your illustration, and then record the total magnification you used. And I'll show you what that looks like. So let's take a look at the lab guide, and we'll see this first part. You can use some of your lecture notes, but here is the components of blood that you can kind of use as your slide at, again, 400 times magnified to do that first illustration in the worksheet. So that's the first activity, is drawing an illustration of the components of blood. You can label the components, the red blood cells. Anything stained darker would be a white blood cell, and then a platelet would be a tinier circle or a thrombocyte. Blood tests in normal values. Let's go through some of these, and then blood types. So let me take us back to the worksheet. So that was our first activity. Answer these questions. Identify the major functions of blood and which cells carry those out. You can use your textbook for that, or Google, whatever resources you want. Identify the functions of plasma. Where specifically are blood cells all produced? I'll give you half the answer. Your red blood cells are produced in the bone marrow of your bones. What are 2 unusual features of red blood cells, and what is significant about these features, and how does structure relate to function? And to answer that question, I'm just going to pull up a PowerPoint showing you a little bit what a red blood cell looks like. I used this in another class. So here are red blood cells, and some things that are unique to erythrocytes or red blood cells, that they do not have a nucleus. It's lost during development. So they're kind of these disc shaped with thick edges. It looks like a donut with a hole in it, and it's biconcave. And the main function is to transport oxygen tissue. And the reason why it doesn't have a nucleus is that it needs to kind of pack as much oxygen as it can into the red blood cells. So the sole purpose of a red blood transport oxygen, why it does not have a nucleus. So that's one unique thing about the structure. Again, another unique thing is that it has a disc shaped with thick edges. So the thick edges kind of maximize surface area as well for transporting oxygen. So hopefully that helps with one of those questions as well, but you can use your lecture notes or any other source for that. Here are blood tests in normal values. So in order to kind of test the number of red blood cells in the sample, we can take a hematocrit, which is the percentage of blood volume comprised of red blood cells, and normal range is about 35 to 50% of a blood sample. Will be that red blood cells, and usually males have a slightly higher hematocrit than females. Total red blood cell count is the total number of red blood cells in 1 millimeter cubed of blood, and you have about 4 to 6 million of red blood cells. Hemoglobin concentration is the concentration of the protein in grams per 100 milliliters. And hemoglobin is important because the amount of hemoglobin in your red blood cells will directly affect how much oxygen they can transport. So if you have too little hemoglobin, or if you're taking a blood test and someone has, let's say, 8 or 10 grams per 100 milliliter, that probably means they're having low oxygen because they don't have enough hemoglobin to transport the blood. Taking total white blood cell count is important because it can be an indicator for disease as well. And normal range is about 4,000 to 11,000 white blood cells per milliliter of blood. And then the total platelet count, here's the normal range. And platelets refer to how well your blood can clot in terms of if there's damage to a blood vessel. So again, if the total platelet count is too high or too low, that could also be an indicator of disease. So these blood tests serve as a diagnostic purpose. In terms of, let's see, this activity, I think uses information as well as the information from your text to answer the questions on page 3. So we'll go back to that. So here are some questions. What is anemia polycythemia? Poly means many. So I'll let you guys research those a little bit on your own. They're different disorders with blood. A female complains of being tired all the time. She also has been dieting. If she's anemic, which of the above tests would you expect to be abnormal? And what is the likely cause of anemia? So I'll let you guys kind of work through that on your own. Once you know what anemia is, you'd be able to tell that's her red blood cells are low, her white So I'll let you work through that as kind of a critical thinking question. A patient has a sore throat and fever for 4 days, which of the above blood tests do you expect to be abnormal? And usually if someone is fighting off an infection or a fever, their white blood cell count will be increased because your white blood cells are the cells that fight off infection. So if this person has an infection, they have a fever, a sore throat, it sounds like strep throat, their white blood cells have probably risen to try to fight off that infection. A person who feels tired all the time and looks pale might be anemic. Further tests reveals that this person has a platelet count of 60,000 millimeters cubed, 60,000 platelets per millimeters cubed of fluid. What is the specific cause or name of this anemia? So this will be a type of anemia that's related to platelet count. So specifically, if they have 60,000 platelets, where does that fall in the range? That's way below normal range. So you're going to want to search or research anemias with low platelet count, and that would be a specific type of anemia for number 5. So a little bit of research shown there, but hopefully it'll be interesting for you. And then get into blood type, which is probably where people have the most, not problems, but it's a little confusing talking about blood type. We're going to kind of go through it as simply as I can. A person's blood type is based on the presence or absence of antigens. And the antigens are A and B, and they will be on the surface of your red blood cells. These antigens will elicit an immune response, like producing antibodies. Antigens are typically proteins. Because there are 2 antigens involved in the ABO blood group combination. So let's just take a look at this type here. If you're type A blood, you have the A antigen, which has kind of a circle shape to it. And again, these antigens will always be on the surface of your red blood cells. In your plasma, you'll produce an antibody against the B antigen. So that if you look at this right here, this anti-B antibody has kind of a cube structure to its head so that it'll actually grab hold of this B antigen. If we ever mix type A and type B blood, the antibodies in the plasma are going to actually grab hold of those antigens on the type B red blood cell. And when those antibodies grab hold of those antigens, they're going to cause about now? Can you hear me now, guys? So sorry about that. My internet connection was a little shaky, but I'm back and the recording was going on. So this will be an interesting recording. So the recording. In our blood types, we have antigens on the surface of our red blood cells. So I'll just talk about myself. For example, I'm type A. So I have A antigens on the surface of my red blood cells. And I have the anti-B antibodies in my plasma. And I went ahead and turned off my video to help see if that helps with the recording. These anti-B antibodies would grab hold of antigen Bs on the surface of type B blood cells, so that if we were to ever mix type A and B blood, the antibodies against each other would grab hold of those cells and cause clumping or agglutination. And when that occurs, that means a bad blood transfusion has occurred. So your blood type always refers to what antigens are on the surface. If you have type A blood, you have the A antigen on the surface. Type B blood has the B antigen on the surface. If you have type AB blood, you have both antigens on the surface of your red blood cells. Because you have both antigens present, you're not going to create any antibodies against those antigens because you don't want to attack your own cells. So type AB people are called the universal receivers of blood because they have no antibodies in their plasma. You can give them whatever blood cells you want because their antibodies won't attack it at all. And then type O does not have any antigens on its surface. Type O blood, because it's basically, I call it the naked blood cell. It has no antigens on its surface. So type O blood is considered the universal donor. When you donate your blood, you're only donating your blood cells. So when we donate type O blood, type O people have both antibodies in their plasma. Donate type O blood, you're just donating these naked red blood cells. So type O red blood cells, they're naked. They can go to any person, any blood type, because no antibody would be able to grab hold of them to cause an agglutination. Type O people, though, they do have antibodies against both types of antigen in their plasma. So if you are a type O person, you can only receive other, I'm calling them naked red blood cells, other red blood cells from type O people, because your antibodies would attack or grab hold of any other red blood cell of any other type of blood because you have both antibodies in your plasma. So this chart is actually really nice to describe that. So these are the four main types of human blood. They're genetically determined. However, genetics involved will not be addressed until you complete your genetics laboratory activity later, which we're not going to do. And this kind of describes the antibodies and how they grab hold of opposite antigens. When that happens, they clump together, causing agglutination of the red blood cells, meaning your red blood cells will clump together and not be able to travel throughout the body to deliver the oxygen to the tissues, completely destroy the cell, causing hemolysis or the splitting of that red blood cell. So this is a look at how the antibody binds to an antigen. So again, the antibody itself has the shape of a perfect antigen that fits inside of it. And when that occurs, when it can grab hold of that antigen, that's what causes clumping. And you can see this occurring here. The antibodies have kind of clumped these cells together, and that inactivates all the cells. The last part, another important factor to consider is the RH or the D antigen. So everyone is positive or negative. They either have this RH factor, which is just the D antigen. So it's just an extra antigen that you either have or you don't have. And if you have it, you're positive. And if you don't have it, you're negative. So everyone is either RH positive with this D antigen on its surface, or they're RH negative with no antigens. And in the general population, most of us are actually positive, and about 14% are negative for this RhD antigen. And this factor must also be considered in blood transfusions due to the aforementioned potential for agglutination. And I'm going to talk a little bit about this RH factor by showing you this PowerPoint, and I'm going to describe why this is important in pregnancy. Because the RH factor, if you have it and you're positive, it's a little different because in the RH blood group, if you're positive, it means you have that antigen, but you can only form antibodies against that RH positive antigen, or antibodies will only develop if an RH negative person is exposed to RH positive blood. To the blood and can cause some incompatibility in pregnancy. So for example, if you're a mother and you're pregnant and you're RH negative, so you don't have the RH factor, if your fetus is RH positive, the blood can actually mix during delivery so that the mother might produce antibodies against that fetal blood, the RH factor. This usually doesn't occur, or it doesn't cause a problem in the first pregnancy. So I'm going to zoom in on this picture a little bit more. So here we have a first pregnancy. During labor and delivery, normally blood does not cross the placenta, but during labor and delivery, blood can cross the placenta. And when that happens, the maternal or the mother blood can form antibodies against the So then in subsequent pregnancies, now you can see this mother has the antibodies present against the Rh factor, so that if it has another Rh positive fetus, what happens is antibodies can cross the placenta. So blood normally, red blood cells do not, but antibodies you can see can cross the placenta. So if the mother has another Rh positive fetus, now she has antibodies built up against it. And these antibodies can cause clumping of the fetal red blood cells, and that can cause what we call hemolytic disease of the newborn, which can be fatal to the newborn. It can cause lysis or splitting apart of its cells and cause death. And to avoid situations, a mother can be given a shot that kind of doesn't allow her antibodies to work across the fetus. So that's how that Rh factor has to do with pregnancy. And hopefully that made sense a little bit. And let me take us back to where we are in your lab guide, blood typing now. Okay, so I think this is part 3, the blood type activity. And let's make sure we are here. So I'm going to kind of show you, let's take you through, this will be a fun. Let's start with and let me know what you guys can see here. Let's take us through the lab guide. So here are the materials to complete the blood typing activity in this lab. And what we're doing here is we have 4 synthetic types of blood. So we need to determine the blood type of these 4 blood samples. And to do that, we are going to use different antibodies. So here's antibody A, antibody B, antibody D, which will give us the positive or negative factor. And what we're going to do is we're going to put antibodies in these we're going to add antibodies to each of the 4 different samples. So we'll run through this 4 times. And if we see kind of clumping or agglutination occurring, that means that the antibody, for example, antibody A, if it clumps when we put it into a well with a synthetic blood type, that means that that antibody A has found the A antigen. It's causing clumping of the blood, and that means that this blood is type A. If the B antibody causes clumping, it means that that B antibody has found the B antigen, and clumping will then tell us that that type of blood is type B. And if we see clumping in the D, the Rh factor, then that means that that person will be positive for that antigen. And I'll show you what I mean here. So we're placing 2 or 3 drops of the synthetic blood sample. It's placed in each well, and we're labeling the wells A, Rh, and B to stand for the A antigen, the B antigen, and the Rh antigen. Then we're placing antibodies in each of the wells. So we're placing the blue antibody into the antigen A well, the anti-B antibody into the antigen B well, and the RH antibody into the RH well. And again, what we're doing here is we're just adding antibodies to the samples of blood. And if the antibodies find an antigen to grab hold of, and again, the antigen will be what it's that antibody is named for, it will cause clumping. So if we see clumping, and I'll show you what that picture is in each of these wells, that clumping will be the blood type. So sample placed on a light box for easier viewing, and then the images of the results are on the subsequent pages. So here's sample 1 results. So here we have a normal result. So this is a person, sample 1. Let's say this is Joe. This is Joe's blood results. And we're trying to put a blood type on the blood of Joe. So what we have here is we see clumping in the antigen A well. So what this means is that when we put that antibody against A in this well, it found the A antigen to clump hold of. So we know that sample 1, Joe, his name is, let's call him, he has the A antigen on the surface of his red blood cells because it caused clumping here. The antibody found that antigen. So Joe, sample 1, is type A. Is he type B? And is he positive or negative? And again, agglutination gives us a positive reaction, and no agglutination means that antigen is not present. B is not present. When we put the anti-B antibody in the B well, it didn't find B antigens. And when we put the RH antibody, it wasn't able to find that RH factor. So sample 1, blood type, he is A negative. Then you'll go through this 3 more times with sample 2, sample 3, and sample 4. And again, if you see agglutination, in this one, I think it's just showing agglutination in B. Wherever you see agglutination, that will give you the blood type. RH is a little hard to see, but it's becoming gelatinous, and it's not agglutinating. Here's sample 3 results. It looks like it's agglutinating in A and B. And sample 4 results, it doesn't look like there's agglutinations in any. So I'm going to see if you guys can do that part on your own. You're going to put all your results here. You're going to give the blood type and then put if it's positive or negative. And it takes you through again how to do this. So in this example of the blood type, only the A shows agglutination, indicating the presence of that A antigen. So this person is A negative because the RH did not agglutinate. All right, so and then you have one final question to do. And again, if you're here live with me, I'll answer any questions with you guys after I stop the recording. Let's say you're in an emergency room and your patient has lost a lot of blood. Their blood type is AB positive. There's a shortage of blood transfusions and all the ER staff can find is O negative. And based on what you have learned about blood types, is it safe to give this person a transfusion with blood? And explain your response. Well, O negative is considered the universal donor because it doesn't have any antigens on the surface of its cells. Remember, O is our naked red blood cell. So because it doesn't have any antigens, those red blood cells can be given to anybody because no matter what, there will be no antibodies that can grab hold of it and cause clumping. So this is a safe transfusion because you can give O negative, it's the universal donor. You can give it to anybody because it does not have antigens on its surface. So I'll go ahead and stop the recording and I'll answer any questions for those of